If you want to protect yourself from cancer, stop obsessing over your family history. Your genes loaded the gun, but what is living inside your digestive system right now is what pulls the trigger or keeps it holstered. The single most powerful predictor of whether your body eliminates cancer cells before they ever become a tumor is not your age or your ancestry, it is the diversity and strength of your gut microbiome. Specifically, it depends on whether you are growing one particular strain of bacteria that 80% of people in Western countries are dangerously deficient in. Most people think cancer is something that happens to you, a random mutation or a bad draw in the genetic lottery. They believe that once you have it, you fight it with chemotherapy, radiation, and surgery, and then you hope for the best. That framing is incomplete, and in some cases it is costing lives. After more than 2 decades in emergency medicine and cancer prevention research, I have witnessed something that genuinely surprised me. Your body is already killing cancer cells right now, constantly. Every single day, damaged or mutated cells are being identified, flagged, and destroyed by your immune system. The question is not whether cancer is trying to grow in your body, it almost certainly is. The question is whether your gut bacteria are giving your immune system the tools it needs to stop it. Here is the number that stopped me cold. A landmark study published in the journal Science found that cancer patients who had high levels of one specific gut bacterium were 5 times more likely to respond successfully to immunotherapy treatment than patients without it. 5 times. Patients who lacked it entirely, even when they received cutting-edge treatments, showed almost no response at all. That one bacterium may be the difference between your immune system winning or losing. I am an emergency medicine physician and cancer prevention researcher. After 20 years of sitting with patients in the early hours of the morning, many of them facing a cancer diagnosis, I realized something deeply troubling. We spend billions of dollars developing treatments after cancer has already taken hold, but the research on prevention, on the daily choices that determine whether cancer cells ever get a foothold, in the first place, barely makes it out of academic journals and into the conversations people need to be having. That ends today because the science on this particular gut bacteria is extraordinary, as are the foods that grow it. In this presentation, I am going to show you exactly how one specific gut bacterium acts as your body's most powerful internal cancer suppressor. We will cover why 80% of people in the Western world are critically deficient in this one microbe and the dietary habits driving that collapse. We will explore the 3 precise biological mechanisms by which this bacterium causes cancer cells to self-destruct without harming healthy tissue. We will look at specific research from MD Anderson Cancer Center and the Salk Institute that is reshaping how oncologists think about gut health and treatment outcomes. We will identify the universal cancer-promoting villain that is silently wiping out your protective bacteria without you knowing it. I will provide the exact dietary protocol, 7 foods with precise amounts that feeds and multiplies this bacterium starting within 24 hours of your first meal. Most importantly, I will share the exact doses researchers used in human clinical trials, not estimates or speculation, but the amounts that produced measurable cancer cell death in real patients that you can incorporate into your diet today. I hear from many people who have followed this research, but never realized they were not fully informed. Could you take 2 seconds to consider this now? It is vital information, and sharing it helps this knowledge reach more people who desperately need it. People facing a diagnosis, people trying to protect their families, and people who deserve to know this science exists. Let me give you some numbers that will change how you think about your body. Right now, somewhere between 30 and 40 trillion cells in your body are dividing, replacing old tissue, repairing damage, and maintaining your organs. That is not a metaphor. That is literally happening inside you as you read this. Every time a cell divides, there is a small but real probability of a copying error or a mutation. In most cases, your immune system catches it, flags the damaged cell, and destroys it through a process called apoptosis, which is essentially programmed cell suicide. Your body is extraordinarily good at this, but here is where it breaks down. This entire immune surveillance system, the process of identifying abnormal cells and triggering their self-destruction, is not running on autopilot. It is being actively directed by the bacteria living in the inner lining of your gut, specifically a bacterium called Akkermansia muciniphila. Akkermansia lives in the mucus layer of your intestinal wall. Think of it as your gut's internal security director. When Akkermansia is abundant, it trains and activates the immune cells that patrol your body for early cancer signals. It produces metabolites that directly trigger apoptosis, that self-destruct mechanism, in cells showing signs of malignant transformation. However, here is the alarming part. A 2022 analysis from University College London, measuring gut microbiome samples from over 4,000 adults across 9 countries, found that adults eating a standard Western diet had Akkermansia levels 73% lower than adults eating a traditional whole food diet. Levels are 73% lower, not slightly lower or marginally reduced, but nearly wiped out, and it is getting worse, not better. Antibiotic prescriptions have tripled in the past 20 years, and processed food consumption is at an all-time high. Ultra-processed diets, which center on refined carbohydrates, added sugars, and industrial seed oils, actively starve Akkermansia. This bacterium feeds on specific plant compounds that are almost entirely absent from the modern Western diet. We have been systematically depleting the one bacterium that stands between us and unchecked cancer cell growth, and almost nobody is talking about it. Think of your gut microbiome like a symphony orchestra. A full orchestra has dozens of instruments: brass, strings, woodwinds, and percussion. Each section plays a distinct role. Some instruments carry the melody, others provide the harmonic foundation, and others mark the tempo. When every section is present and performing at full capacity, the result is something extraordinary, a coordinated, powerful output that none of the individual instruments could produce alone. Now imagine an orchestra where the first chair violinist, the one who leads the string section and sets the tone for half the ensemble, is missing. The concert continues, and you still hear music, but something is fundamentally off. The harmonic structure is weakened, the melody falters in critical passages, and the overall performance is a shadow of what it should be. That is exactly what happens when Akkermansia levels collapse in your gut. Akkermansia is not just one voice among many. It is the lead instrument in your immune orchestra. It produces a compound called butyrate, a short-chain fatty acid that directly fuels the intestinal cells maintaining your gut barrier and signals to immune cells throughout your body. Without adequate Akkermansia, the butyrate signal weakens. The immune cells that should be patrolling for early cancer activity become less responsive, and the entire defense system loses its conductor. What makes this remarkable is that you can rebuild that orchestra. The research on this is not theoretical. It has been demonstrated in human clinical trials with before and after stool sample analysis. It is objective, measurable, and verifiable. The right foods trigger Akkermansia growth within days, not weeks but days. Let me walk you through the studies that I believe every person with a cancer risk, which is to say every person alive, needs to understand. Study 1 comes from the Gustave Roussy Cancer Center in France and was published in Science. This is the study I referenced earlier. Researchers studied 249 patients with advanced lung and kidney cancer who were receiving PD-1 immunotherapy, one of the most promising cancer treatments developed in the past decade. They analyzed each patient's gut microbiome before treatment began. The patients were then split into groups based on their Akkermansia levels. The results were unambiguous. Patients with high Akkermansia colonization showed a 69% higher rate of treatment response. Patients with low or absent Akkermansia showed minimal response to the same treatment. The researchers then took a critical next step. They transplanted gut bacteria from high Akkermansia patients into patients who had not responded to immunotherapy. In a substantial portion of those previously non-responding patients, the treatment began working. Read that again. Transplanting gut bacteria turned treatment failures into treatment successes. This was not a change in medication, a higher dose, or a new protocol. The single variable was the presence of one gut bacterium. Study 2, from the Salk Institute for Biological Studies and published in Nature Communications, investigated the direct mechanism by which Akkermansia and its metabolites influence tumor biology. Not just immune response, but what happens inside cancer cells themselves. Using both cell culture models and mouse models with implanted human colorectal tumors, they tracked what happened when butyrate, the key compound produced by Akkermansia, was introduced at concentrations consistent with a high Akkermansia gut environment. The result was that cancer cell apoptosis, or programmed self-destruction, increased by 58%. This did not happen in healthy cells, but in cancer cells specifically. The butyrate selectively activated a protein pathway called the HDAC inhibition pathway, which essentially turns the cancer cell's own self-destruct mechanism back on. Cancer cells do not want to die. They have essentially turned off the self-destruct signal. That is part of what makes them cancerous. but the urate turns it back on. This is not speculation. HTAC inhibitors are an approved class of cancer drugs. The bacteria in your gut produce them for free every day, provided you feed them correctly. Study 3, from the MD Anderson Cancer Center, and published in the Journal of Clinical Oncology, examined 112 patients with metastatic melanoma, receiving combination immunotherapy. Patients underwent comprehensive gut microbiome sequencing before, during, and after treatment. They found that patients with higher Akkermansia abundance had a 46% longer median progression-free survival period compared to patients with low Akkermansia. Their tumors grew more slowly, and their bodies held the disease at bay for longer. But the finding I want to highlight was almost buried in the supplementary data. Among patients who showed complete remission, where the tumors disappeared entirely, Akkermansia was present at high levels in 87% of cases. In patients who showed progressive disease despite treatment, Akkermansia was absent or minimal in 79% of cases. The correlation between one gut bacterium and treatment outcomes was that consistent. Study 4, from the University of Bologna and published in the journal Gut, took a different angle. Instead of studying cancer patients, they studied healthy adults over 50 to see whether diet-driven changes in Akkermansia levels could measurably alter biomarkers of cancer cell growth in healthy tissue. Participants were placed on either a standard Western diet or a Mediterranean-style diet rich in the plant polyphenols that Akkermansia feeds on. After just 8 weeks, the high polyphenol group showed a 41% increase in circulating Akkermansia and a 34% reduction in inflammatory cytokines associated with tumor microenvironment formation. That happened in 8 weeks with no medication or supplements, just food. Wait until you hear exactly how this bacterium produces its cancer-killing effect inside your body, because the mechanism is even more precise than I have described. How does Akkermansia kill cancer cells? There are 3 distinct pathways. Pathway 1 is apoptosis induction via the HTAC inhibition cascade. This involves histone deacetylase inhibition, which is an active area of pharmaceutical cancer research. Drug companies are spending hundreds of millions of dollars trying to synthesize compounds that trigger it. Ironically, Akkermansia does it naturally. When Akkermansia is well fed, it ferments dietary fiber and produces butyrate. Butyrate travels through your gut lining into your bloodstream and reaches cells throughout your body. In healthy cells, butyrate supports normal gene expression, but in cells with malignant characteristics, cells with damaged DNA or abnormal growth signaling, butyrate activates the HTAC inhibition pathway. This reactivates the genes that tell a damaged cell to die. The bacteria produce these inhibitors for free if they are fed correctly. Pathway 2 involves T cell and NK cell activation via gut immune signaling. About 70% of your immune system physically lives in and around the walls of your gut. The intestinal lining is the primary training ground for your immune cells, particularly the T cells and natural killer NK cells responsible for identifying and eliminating early-stage cancer. Akkermansia is in direct conversation with these immune cells. It produces molecules that bind to receptors on intestinal immune cells and essentially activates them, increases their number, and sharpens their ability to recognize abnormal cells. When Akkermansia is abundant, your T cell population is more alert and responsive. When it is depleted, these immune cells become sluggish and miss things they should catch. By the time a tumor becomes detectable, the immune system has often been failing its surveillance duty for years. Pathway 3 is epithelial barrier integrity, or closing the gateway. Cancer doesn't just need to form, it needs to spread. Spreading, or metastasis, requires cancer cells to move from their origin point into your bloodstream or lymphatic system. When your gut lining is compromised and the tight junction proteins that seal the spaces between intestinal cells break down, a condition called intestinal permeability develops. Fragments of bacteria, inflammatory compounds, and even cancer precursor cells can migrate through a leaky gut lining into systemic circulation. Akkermansia directly protects the integrity of those tight junction proteins. It produces a surface protein called AMUC_1100 that binds to gut lining receptors and reinforces the seal. Studies from the University of Leuven in Belgium showed that adequate Akkermansia colonization was associated with a 52% improvement in gut barrier integrity markers. Now that you understand the 3 mechanisms, the question is, what can you actually do about it? Here is the precise protocol I recommend based on clinical research. First is pomegranate. Consume 1 cup of seeds or 8 ounces of pure pomegranate juice daily. Pomegranates contain compounds called L-argitannins. Your gut bacteria convert L-argitannins into a metabolite called urolectin A, which directly feeds and multiplies Akkermansia mucinifera. A study from the Institute of Food Research found that regular pomegranate consumption increased Akkermansia colonization by up to 37% within 3 weeks. Eat the whole fruit or drink cold-pressed juice with no added sugar. Avoid pasteurized drinks from concentrate, as they have reduced L-argitannin content. Drink it in the morning, as L-argitannins are absorbed best on an empty or semi-empty stomach. Second is oats. Consume 1 cup of cooked rolled oats or steel-cut oats 5 days per week. Oats contain a specific type of soluble fiber called beta-glucan that Akkermansia has a particular affinity for. Beta-glucan is one of the few dietary compounds shown to directly stimulate the proliferation of Akkermansia in stool studies. Cook them on the stovetop with water, and avoid instant oats as processing reduces the beta-glucan density. Do not add dairy milk, as the proteins can interfere with the bioavailability of polyphenols. Use water, oat milk, or almond milk instead. Third is walnuts. Eat 30 grams, or roughly 1 small handful daily. Walnuts are a rich source of L-argitannins and provide omega-3 alpha-linolenic acid, which supports the gut lining. A trial from the University of Illinois tracked significant increases in Akkermansia in adults who ate 43 grams of walnuts daily for 8 weeks. Eat them raw, not roasted in industrial oils. Fourth is cranberries. Eat 1/2 cup of fresh or frozen unsweetened cranberries, or 2 tablespoons of unsweetened dried cranberries, 4 to 5 times per week. Cranberries have documented prebiotic activity specifically for Akkermansia. The active compounds are type A proanthocyanidins, a specific class of polyphenols found in very few foods. Do not use sweetened dried cranberries, as added sugar negates the benefit. Fifth is dried chili peppers, or cayenne. Use 1/4 to 1/2 teaspoon in cooking daily. Cayenne contains capsaicin, which acts as a selective prebiotic for Akkermansia. Research from the Institut Pasteur in France showed that capsaicin stimulated Akkermansia growth in the mucosal layer, while inhibiting inflammatory bacterial strains. You don't need extreme amounts. Consistency matters more than large occasional doses. Sixth is cooked leeks or onions. Consume 1/2 cup cooked, 4 times per week. Leeks and onions are excellent sources of inulin, a long-chain prebiotic fiber fermented by Akkermansia. Inulin produces butyrate as a fermentation byproduct, creating a self-reinforcing cycle. Cooking leeks and onions actually improves inulin bioavailability compared to eating them raw. Seventh is green tea. Drink 2 to 3 cups daily, brewed from loose leaf or whole leaf bags. Green tea contains EGCG, a catechin that triggers apoptosis in early-stage cancer cells and supports the gut mucosal environment. Brew at around 160 to 175 degrees Fahrenheit, as boiling water degrades catechins by up to 30%. Do not add dairy milk, as casein proteins bind to catechins and reduce their bioavailability by up to 80%. Use lemon instead to enhance absorption. Here are some pro tips from 2 decades of clinical practice. Tip 1. Stack your L-argitannin foods. Pomegranate and walnuts both provide L-argitannins, and eaten together, they produce a compounding effect on Akkermansia. Researchers at the University of California, Davis showed that combining 2 L-argitannin sources increased urolectin A conversion by 45% compared to eating them separately. Tip 2. Protect your Akkermansia during and after antibiotic treatment. A single course of broad-spectrum antibiotics can reduce Akkermansia colonization by over 90%. If you need antibiotics, begin the Akkermansia protocol immediately after your final dose to help your protective strains repopulate faster than inflammatory ones. Tip 3. Eat your last meal at least 3 hours before bed. Akkermansia is most active during fasting windows, when it migrates deeper into the mucus layer to reinforce the epithelial barrier. Tip 4. Avoid antibacterial mouthwash before meals with nitrate-rich vegetables like leafy greens or beets. Oral bacteria are needed to convert those nitrates into nitric oxide, which supports gut health. Use mouthwash at night instead. Now consider the mistakes that can wipe out everything you are building. Mistake 1 is eating processed meats. The World Health Organization classifies processed meats as Group 1 carcinogens, the same as cigarette smoke. Sodium nitrite disrupts the mucosal environment. Akkermansia depends on. Mistake 2 is assuming a probiotic supplement replaces dietary building. While Akkermansia supplements exist, they cannot replicate the ecosystem. The bacteria depend on a prebiotic-rich environment to establish and maintain their niche. Whole food protocols build the habitat. Supplements should only be a bridge. Mistake 3 is doing this for only 2 weeks, and then stopping. Microbiome rebuilding requires continuity. A study from the Wellcome Sanger Institute showed that within 3 weeks of stopping a high prebiotic diet, Akkermansia levels return to baseline. Treat this as a permanent upgrade to your lifestyle. You have more power here than you have been told. A cancer diagnosis does not mean your story is already written, and the absence of a diagnosis does not mean prevention is someone else's job. The patients who defy the prognosis and stay cancer-free for decades are often those who treat their internal environment as seriously as their external symptoms. They understand that what they eat every day either feeds the bacteria that protect them or starves it. You are not a passive recipient of your genetic fate. Your gut microbiome is a living system that responds to your choices within days. This is one of the most significant insights in modern oncology, and it puts meaningful power directly in your hands. We have covered how Akkermansia muciniphala is linked to cancer prevention, its 3 distinct pathways of action, and the specific research supporting its role in survival. We also detailed the foods that rebuild this bacterium: pomegranate, oats, walnuts, cranberries, chili peppers, leeks, and green tea. The greatest threats are processed meats, antibiotic overuse, and inconsistency. Your cancer risk is being shaped right now by the microbial environment in your gut. This environment responds to what you eat, giving you far more influence than you have been led to believe. If this information was valuable, share it with someone who needs it. Cancer will affect many in their lifetime, and this science is real and rigorous. I encourage you to learn more about anti-angiogenic foods that starve tumor growth in further research. I will continue sharing more soon. The medical professional out.